Assalamu alaikum friends. Hope so that you all will be fine. In this video, I am going to discuss about a very simplest topic of glycogen metabolism, that is glycogenesis. Okay, friends, before starting our lecture, it's my humble request to you that if you like this video, then kindly subscribe to my channel and share. Okay. So let's start our lecture with the definition of glycogenesis. Glycogenesis means synthesis of glycogen from glucose. The synthesis of glycogen from glucose, it is the definition of glycogenesis. Now coming toward the site for the process of glycogenesis. This process occurs in liver and muscles. Okay, and the organelle that is required, that is cytosol, that is cytosol. Okay, friends, this glycogenesis process occurs in well-fed condition, and there is important hormone insulin that is secreted from beta cells of islets of Langerhans. It is responsible for the synthesis of glycogen. Now coming toward the steps of glycogenesis. Steps. Steps of glycogenesis. So let's see steps. First of all, glucose, it converts into glucose 6 phosphate by action of glucokinase and hexokinase enzyme. This glucokinase enzyme acts only in on liver, while hexokinase enzyme, it acts on muscles, and this reaction requires magnesium ions. Okay. In next reaction, this glucose 6 phosphate by action of phosphoglucomutase enzyme, it converts into glucose 1 phosphate. This mutase enzyme is responsible for shifting of phosphate group from carbon number 6 to carbon number 1. In next reaction, by action of UDP glucose, sorry, UDP glucose pyrophosphorylase, and here there is a release of PPI, that is pyrophosphate. One phosphate, this phosphate released, and from here this only one phosphate released, and they both release in the form of PPA, that is pyrophosphate. One phosphate from here and one phosphate from here, and there is a formation of UDP glucose. Okay, friends, this is called as, this is the first step in glycogenesis, that activation of UDP glucose. Glucose converts into the glucose 6 phosphate, and with the help of phosphoglucomutase, it converts into glucose 1 phosphate, and it also requires magnesium ion. In next reaction, glucose 1 phosphate converts into UDP glucose by action of UDP glucose pyrophosphorylase. One phosphate released from here and one phosphate released from here, and both are released in the form of PPI, that is pyrophosphate. Okay, this is the first step. This is the first step. Now, for the synthesis of glycogen, there is need of glycogen primer. What is glycogen primer? It is a pre-existing molecule to which there is attachment of glucose. It is a pre-existing glycogen molecule to which there is addition of glucose molecule. In absence of glycogen primer, there is a substance that is glycogenin. It is a protein with the oligosaccharide side chains. Okay. Now, glycogenin, it is very important for the synthesis of glycogen. In absence of glycogen primer, this substance is used. It is a protein with the oligosaccharide side chains. Okay, this is the second step. This is the second step, that is, the first step is activation of UDP glucose. The second one is initiation of glycogen synthesis. This is the second step, initiation of glycogen synthesis. Now, in next reaction, what happens? That there is a successive attachment of glucose molecule here, and the important enzyme that performs this action, that is glycogen synthase. There is a successive attachment of glucose molecule that helps in the elongation of this chain, and approximately 13 UDP glucose molecule they are used and UDP released, while glucose molecule they get attached here and are very important in the elongation of chain. Okay, now let's see here. This is the glycogenin. Sorry. Okay, glycogenin. Okay, this is the third step, which is called as elongation. The first step is activation of UDP glucose, second step is initiation of glycogen synthesis, and third step is the elongation. Now, the fourth step in next reaction, I'm writing here. In next reaction, there is use of branching enzyme. When this glucose chain elongates up to 12 to 13 glucose residues, then approximately 6 to 8 glucose molecule they gets detached from here and gets attached to the here. In this way, branching formation takes place. Okay. Now here there is a glycogen. 6 to 8 glucose molecule they gets detached from here and gets attached to another point. Okay, let's see here. This is glucose. Okay. Now here, this bond, this is the alpha 1 4 glycosidic bond. This is the alpha 1 6 glycosidic point. This is a branch point, alpha 1 6 glycosidic bond. Now, this is the fourth step. This is the fourth step. This is the called, which is the branching formation. The first step is activation of UDP glucose. Second is initiation of glycogen synthesis. Third one is elongation. Fourth one is the branching. Now, this third and fourth step, they are repeated again and again. Mean elongation. Fifth step is elongation and branching. They are repeated again and again, ultimately until they lead to the synthesis of glycogen. The sixth one is the glycogen synthesis. Glycogen. Until they lead to synthesis of glycogen. So these are the steps of the glycogenesis. The first one is activation of UDP glucose. Second one is initiation of glycogen synthesis. Third one is elongation. Fourth one is branching. Fifth one is elongation and branching. They are repeated again and again until the last one, the synthesis of the glycogen. Here is the important enzyme is glycogen synthase. It is a very important enzyme, and insulin activates this enzyme. And in well-fed condition, the glycogenesis process takes place with the help of hormone, that is insulin, which is released from the beta cells of islets of Langerhans. Okay, friends, this was all about the glycogenesis. Glucose converts into glucose 6 phosphate with the help of this mutate enzyme. It converts into glucose 1 phosphate with the help of UDP glucose pyrophosphate phosphorylase. There is a release of PPI pyrophosphate, 1 phosphate from here, 1 phosphate from here, and collectively there is a pyrophosphate. There is synthesis of UDP glucose, and this is the first step, activation of UDP glucose. Now, for the synthesis of glycogen, there is a need of glycogen primer, but in absence, this glycogen is used. It is a protein with oligosaccharide side chain. This is the second step, initiation of glycogen synthesis. Now, next step, with the help of this enzyme, glycogen synthase enzyme, approximately 12 to 13 glucose molecules they get attached to this side and leads to elongation of this chain. Okay, this is third step, elongation, and the fourth one is branching. With the help of branching enzyme, there is a formation of branching. Here you can see there is a branching formation, and there is a formation of bond. This alpha 1 6 glycosidic bond. This is alpha 1 4. This is alpha 1 6 glycosidic bond, and approximately 6 to 8 glucose molecule gets detached from here, and they get attached to another point, and there is formation of branch. And elongation and branching, they repeat it again and again until they lead to synthesis of the glycogen. So it was all about the glycogenesis. Keep watching and thank you.